As with all processes we use to treat water, it's important to understand the fundamental mechanisms and kinetics that govern those processes. In considering UV disinfection, the same thing is true. And so in this lecture, we'll address the mechanisms and kinetics that we use to describe and understand how microorganisms and viruses are inactivated by exposure to ultraviolet radiation. UV disinfection is a process that has a wide range of applications, including drinking water production, wastewater disinfection, disinfection of wastewater that's going to be reused, and disinfection of air and surfaces, among other things. The reason that UV is used in such a broad range of applications is that the mechanism of inactivation allows microbial and viral inactivation across a wide range of applications. The mechanism of inactivation specifically involves photochemical damage to critical biomolecules. Those biomolecules include the nucleic acids, DNA or RNA, and proteins. Considering first the nucleic acids, and specifically DNA, this is a common image that you can find on the internet and elsewhere that illustrates the process. The basic idea is that a photon will be absorbed by the DNA strand, which leads to dimerization within the DNA strand. For the microorganism to be able to reproduce, it needs to be able to replicate its DNA. This type of damage makes it more difficult for that microorganism to replicate its DNA. If it can't replicate its DNA, then it can't reproduce and it can't cause infection. So this is just a schematic illustration. The graph in the lower right illustrates measured absorbance spectra for nucleic acids that have been extracted from a bacterium, Vibrio harvii, a protozoan parasite illustrated by the red line, Cryptosporidium parvum, an algal culture, Tetraselmis, indicated by the green line, and SARS-CoV-2, the virus that's responsible for COVID-19, by the blue line. You'll notice that there are small differences among these spectra, but basically they all show a peak in absorbance in the vicinity of 260 to 265 nanometers, and then shouldering on either side of that peak. At wavelengths below about 230 nanometers, you start to see strong absorbance taking place again. For proteins, the story is not quite as clear or as consistent. There's actually quite a variation in absorbance behavior among proteins because of the wide variation in their structures. But what we generally observe is that for wavelengths below about 230 to 240 nanometers, the absorbance starts to increase rapidly, moving toward the lower or shorter wavelengths. The most basic model that's used to describe the kinetics of UV disinfection is the so-called single event model. The basis of this model is that we assume that one unit of photochemical damage is sufficient to inactivate the target microorganism or virus. For simplicity in developing this model, we'll assume that we have a monochromatic source of radiation. In other words, one that emits only at one wavelength, like a low-pressure mercury lamp. The kinetics of inactivation are simulated using the same logic we use to describe the kinetics of a pure photochemical reaction. Specifically, the rate at which the concentration of viable or infective microorganisms within a population changes as a function of time is described by the negative of the product of the inactivation constant, K, the fluence rate, E, and the concentration of those organisms. In other words, this is a pseudo first-order process. And in this equation, again, E represents the fluence rate or irradiance. As mentioned in a previous lecture, this is a description of the availability of photons to participate in this process. Mathematically, we can rearrange this equation and then integrate to yield the following form. If we then recognize the formal definition of the UV dose as the time integral of the fluence rate field applied to the target, then we can develop the common form of the single event model, which tells us that we should expect first order loss of a microbial population with an inactivation constant K, and again, dose represents the master variable in describing the behavior of this system. The single event model has logic and mathematical derivation that are direct analogs of the Chick-Watson model. And again, to emphasize, dose is the master variable. So let's examine the use of the single event model for simulation of UV disinfection kinetics. Does it work is really the question. And the answer is, for many microbial and viral populations, yes, the single event model works well. So here's an example of a paper that was published in 2018 in which the infectivity of the murine norovirus, or MNV, as indicated by the triangles here, is described as a function of the applied or delivered UV dose. As we see in this representation, in which the vertical axis is presented on a log scale, we observe first order inactivation to be evident over 4 or 5 log units of inactivation for the murine norovirus. Now this turns out to be important because noroviruses are responsible for a large fraction of human illness that's associated with waterborne and foodborne diseases. So this suggests then that the single event model will do a good job of describing the intrinsic kinetics of inactivation of the noroviruses, and moreover that we can get effective inactivation of the noroviruses at fairly modest doses on the order of maybe 30 or 40 millijoules per square centimeter. Also indicated in this graph is the behavior of MS2, a so-called coliphage. Phage are viruses that infect bacterial cells. Coliphage have the ability to infect coliform bacteria. MS2 is relevant because it's a common surrogate for human viruses, and in the context of UV disinfection, it tends to be more resistant to UV than most human viruses. So it is a conservative surrogate for the human viruses. In broader terms, we can consider the effects of UV radiation, in this case at 254 nanometers, against a wide range of microbial and viral pathogens. In this graph on the vertical axis are UV doses that are required to accomplish 2 log units, or 99% inactivation, of groups of different microorganisms and other pathogens. The gray bars indicate the range of doses that have been reported for 2 log unit inactivation of bacterial spores. Among these, you'll see that there's a fair amount of variability, and they also tend to require relatively large doses, but still manageable doses of UV radiation. The pink bars illustrate the behavior of many bacteria, and generally the vegetative cells of bacteria are highly sensitive to UV, requiring single-digit doses to accomplish multiple units, log units, of inactivation. The green bars represent the responses of protozoan parasites, specifically Cryptosporidium parvum and Giardia lamblia, and we see that they're very effectively inactivated at really low doses. And then lastly, the blue bars illustrate reported behavior among waterborne viruses. Here you'll see that there is a fairly broad range of responses, with, for example, the adenoviruses being fairly resistant. But generally, we can use UV to inactivate essentially any microbial or viral pathogen. Now, I mentioned previously that the single event model works for some microorganisms, but not for all of them. There are some common deviations that are observed from the single event model. These include the existence of a shoulder, that's illustrated up here following the blue dashed line, and tailing following the red dashed line. These are both common deviations from the behavior that's suggested by the single event model, which is indicated by the red solid line here. So these common deviations include a shoulder, which has been attributed to either repair processes or the requirement for multiple units of damage to cause inactivation. The other common deviation is tailing, which has been attributed to population heterogeneity and the association of microorganisms with particles, or embedding of microorganisms within particles. Now, it turns out that for the shoulder, it's difficult to differentiate repair from the requirement for multiple units of damage. And likewise, in the case of tailing, it's difficult to differentiate population heterogeneity effects from particle association effects. But nonetheless, there are these deviations that are commonly observed. So it would be nice to be able to manage them and be able to predict this behavior. There are models that have been developed to address these common deviations, and what I'd like to do is to quickly review 2 of the common models that are used for this purpose. The first is the series event model, which was developed to account for shouldering. The basis of this model is that some microorganisms, or potentially viruses, are not inactivated until they accumulate multiple units of damage. Moreover, an assumption is made that among a population, a threshold dose can be identified that's required to initiate inactivation. Schematically, that looks like this, and in this schematic, what we assume is that M sub I represents microorganisms with I units of damage. So over here on the left, M sub 0 is an unaffected population. The first unit of damage is indicated here, second unit of damage, and so on. Also included in this model, you'll see the nomenclature H nu, which is included to represent the energy of an individual photon. The basic idea here is that each photochemical event is assumed to follow first-order kinetics. Each of these events can be then described following the first-order behavior, and using that logic, we can then use this model to describe how the concentration of microorganisms with I units of damage changes as a function of the dose that's applied. The right-hand side of this equation tells us that we'll lose organisms with I units of damage following a first-order process, but we'll also gain them following a similar first-order process from microorganisms that have one less unit of damage than I. Continuing with this logic, again we assume that all microbes within the population that have less than the threshold N units of damage will retain viability. So mathematically what that tells us is that we can add up the concentration or the total population of viable microorganisms within the population by simply adding up the concentrations of all microorganisms that have less than the threshold number of units of damage. When we apply this to the differential equations that would apply across all units of damage, this leads to a series of differential equations that ultimately results in the series event model. So let's consider what the series event model implies and graphically what it looks like. This graph was taken from a paper published by Blaine Severin et al. In 1983 that's really sort of a classic to illustrate this model, and it describes the behavior of three different microorganisms. The basic idea here is that the series event model provides a mechanistic explanation for the existence of a shoulder in a dose-response curve, three of which are illustrated in this graph. The first of these is for a bacteriophage, F2, the second is for E. coli, and the third is for a yeast called Candida parapsilosis. Generally, what we observe is that as the threshold N increases, the size of the shoulder increases. And that generally goes along with more complicated microorganisms. As we move from relatively small organisms like viruses to bacteria and then to larger organisms like yeasts, the organisms themselves become more complicated, and it's perhaps reasonable to expect that more units of damage will be required to start to observe inactivation. It should be noted that for the special case of a threshold of 1, the series event model reduces to the single event model. That should make sense both mechanistically and mathematically. If we consider the math of this, the last argument in the series event model itself looks like this. For the special case of n equals 1, that summation reduces itself identically to 1. In other words, that part of the equation reduces to 1, and when that happens, what we're left with is the single event model. Now let's consider another model that can be used to describe tailing behavior and potentially shouldering behavior. The specific model I'm referring to is called the Phenotypic Persistence and External Shielding Model, or PPES. It is a two-population model, and there are many such models that have been presented in the literature. The basic idea with these two populations is that one of the populations, or subpopulations, we'll just call it A, is relatively sensitive to UV exposure. The second subpopulation, we'll call it B, is relatively resistant to UV exposure. You could use other models to describe the behavior of these two subpopulations, but for purposes of this presentation, I've assumed series event and single event models for A and B subpopulations respectively. For each of these subpopulations, we can define an initial concentration, A0 and B0, and the initial concentration of viable or infective microorganisms within the population is just the sum of those initial concentrations. For each subpopulation, we can identify a separate inactivation constant, and the shoulder behavior is simulated using the series event model. By extension, this suggests that the vast majority of what we observe in terms of the shouldering behavior is attributable to the relatively sensitive subpopulation, in this case subpopulation A. Mathematically, the model looks like this. Basically, what we've done is just combine the series event model and the single event model. We use the series event model to describe the sensitive subpopulation. We use the single event model to describe the persistent subpopulation. And so how well does this work? Well, it really depends on the data set. It doesn't work for all data sets, but for some data sets we see this characteristic behavior where there is both a shoulder and some tailing that shows up. So on the panel on the left, we see the response of Bacillus subtilis spores. These are bacterial spores that are commonly used in experiments to characterize the behavior of UV reactors, and they tend to be relatively resistant to UV exposure. Here we see responses of Bacillus subtilis spores at 3 different wavelengths, 222, 254, and 282, and we see that in all 3 cases there's the existence of a shoulder and tailing, both of which can be captured by the PPES model. On the right are replicate experiments that are conducted with a population of E. coli, and we see the same sort of behavior showing up. The model itself is applicable over as many as 8 log units of inactivation. Summarizing then, inactivation of microorganisms and viruses by exposure to UV radiation, especially UVC radiation, is caused by photochemical damage to critical biomolecules, specifically the nucleic acids and proteins. The single event model assumes that 1 unit of photochemical damage will cause inactivation of the target microbe or virus, translating to a first-order behavior. There are many microorganisms and viruses that follow this single event or first-order behavior quite well. However, there are common deviations from single event behavior that have been observed in experiments, and these common deviations include the existence of a shoulder or tailing. The series event model provides a mechanistically consistent explanation for this behavior, and mathematically it also is able to describe the behavior that we observe in those sorts of experiments where shouldering is evident. The PPES model is an example of a 2-population model that can account for the effects of both shouldering and tailing. More broadly, the UV doses that are required to inactivate most microorganisms and viruses are quite small, which allows UV disinfection to function as a broad-spectrum antimicrobial and antiviral process.